So I'm going to talk to you today about one of the most amazing things that I think plants do. They do lots of wonderful things, but they're extremely good at making chemicals. And so here you can see a few examples of useful chemicals that plants make. So up here we have artemisinin, which is made by a plant called wormwood, and this is important because it's anti-malarial. Over here we have Taxol, which is made by the yew tree, which is an anti-cancer drug. Here we have another anti-cancer drug produced by mayapple called etoposide. And this molecule, which I'll come back to later on, happens to be called QS21. I'll explain why later. It's a very complicated molecule. It's produced by a tree that grows in a certain part of Chile called the soapbark tree, or Quiaja saponaria. And it's an immunostimulant, so it's included in vaccines as an adjuvant, so it promotes the immune response. So these are just 4 examples of the probably billions of molecules that plants make. Importantly, around 50% of the drugs in current use are either natural products or natural product inspired. And that's because these molecules that plants and also microbes make natural products as well, they don't just make them for fun, they make them because they're doing something useful. And generally these molecules are doing something to do with survival in the environment and with signalling. So they've been honed by evolution to interact with biological targets and pathways. So that's why natural products are a very rich source of bioactives. And most of these molecules are too complicated to be synthesized in the laboratory using chemistry, with the exception of simple molecules such as aspirin. So even morphine, which is of course used very widely, is still sourced from the producing plant, not by chemical synthesis, because it's too complicated. There are over 400,000 species of higher plant on the planet. We currently have the genome sequences for just a few thousand of those. But as we begin to look into those genome sequences, we can see, using computational algorithms, that those genomes are likely to harbour a huge number of genes which encode the enzymes and pathways of specialised metabolism. The vast majority of those pathways we haven't even discovered yet. We just know that based on computational predictions, it looks like there's a lot going on. So I'm going to give you a bit of historical context now, because I think that's very helpful. So my group works primarily, we work on plant natural products, understanding how they're made, what they do, and mechanisms of metabolic diversification. And we became particularly interested in a large group of natural products that plants make, called the triterpenes. So this is actually one of the largest and most structurally complex groups of plant natural products, with hundreds of thousands of triterpenes reported so far. And we became interested for specific reasons to do with this molecule, adenosine A1, which is only produced by oats. So this is often the case that these specialised metabolites are only produced by certain taxonomic groupings of plants, hence the word specialised. So oats make this molecule adenosine, which is antifungal. These are other examples of triterpenes here from licorice, anti-inflammatory, from cucumber and relatives, anti-tumour, also associated with the bitter taste of wild cucurbits. And then the health beneficial compounds ginsengicides from ginseng, and our old friend QS21 from the Chilean tree. So what these have in common is that they are all triterpenoids. They have a triterpenoid scaffold, but you can see that these scaffolds have been decorated with various different modifications. And my reason for becoming interested in this, and I think this is important when thinking about a scientific career, you can't really plan it. You think you can, but actually you can't. So many years ago, I used to work on plant-microbe interactions, understanding the mechanisms by which pathogens infect plants. And this got me interested in this disease called Tacol disease of wheat. It's caused by a soil-borne fungus with a horrible name, Germanomyces graminis variety tritici. It infects the roots, and it causes lesions to form on the roots, and this stops the vasculature from functioning, and so eventually you get patches of stunted wheat plants, And it's called take-all disease because when it's very severe, it takes all the yield away. There's no effective means of control for take-all. The average yield losses are 5 to 20%, but often with complete failure of the crop under severe disease conditions. And the other problem is that it gets worse. The more years that you grow wheat in succession, it gets worse for the first 3 years or so. And then it declines, which is another very interesting story. But basically, if you want to keep growing wheat, you have to go through this pain barrier of losing quite a bit of your crop because of take-all. So back in 1960, a lady called Elizabeth Turner at the University of Oxford noticed that oat roots produce a fluorescent molecule. So this is really high tech. She's coloured the roots in black. This molecule fluoresces under ultraviolet light. So she could see fluorescence here. And she called this a fluorescent antifungal root tip glycoside because it was antifungal. And it turned out this molecule was adenosine, subsequently named by a group at Nottingham, the Crombies. So in 1984, the structure of adenosine was determined, and it's a triterpene with 3 sugars here. I couldn't find, so there was a husband and wife team, Leslie Crombie and Mary Crombie, but I couldn't find a picture of Mary, so you've just got Leslie. Many years on, we decided to ask, so Elizabeth Turner predicted that this root tip glycoside, which was antifungal, was likely to protect oats against attack by the soil-borne pathogen, the take-all fungus. So we decided to address this, and we took a diploid oat species, so cultivated oats are hexaploid, but that was too complicated for us because we hadn't done much genetics. We took a diploid species because all oat species just about make this fluorescent molecule. Here you can see a seedling on a UV transilluminator, and this is our diploid oat, the wild type, and the tip is fluorescing bright blue. There's a bit of leaching here, but you can see the blue fluorescence here. And what we did was we treated this diploid oat species with a chemical mutagen, sodium azide, which induces point mutations. And then we screened the mutants for loss of this fluorescent molecule. And we had a simple question. We just wanted to know whether or not Elizabeth Turner's prediction was right. Does this molecule provide protection against soil-borne pathogens? So because the fluorescence screen was so sensitive, we identified over 100 mutants in the pathway. And we confirmed that, yes, indeed, if you get rid of this molecule, then the seedlings become susceptible to pathogen attack. So this is the wild type, and you probably can't see very well, but the roots are white and healthy. And this is one of our mutants, and you can see dark lesions on the roots. So we'd answered Elizabeth Turner's question. We published a nice paper in PNAS. And this was important at the time because a lot of people were thinking about chemicals that plants make and what those chemicals might do in terms of protecting plants against pathogens. But they were mainly thinking about chemicals that are induced in response to pathogen attack, whereas this is a preformed chemical that's just sitting there in the soil fluorescing bright blue. And so we published this paper, and we thought that would have been the end of the story, because at that time, nothing was really known about triterpene biosynthesis for any plant species, and oat was not a model plant. And to give you some more context, the first plant genome sequence, which was Arabidopsis, was published in December 2000. We published this in 1999. So this is going back into the Stone Age. So we got over 100 mutants. We knew nothing about the pathway. We knew the pathway was likely to be complicated. It was going to be a multi-enzyme pathway. And we reckoned it would involve at least 10 different steps. And although we hadn't cloned any of the genes, the genetics was telling us that the loci that we defined by mutation were not segregating away from each other. They were behaving as if they were a large multi-gene locus. So this meant we carried on with oats, which was a bit reckless at the time. We started to clone the genes based on various types of approaches. And we built, at that time, our own back library, a large insert library. You can now just get those made commercially. This is a snapshot of where we'd got to halfway through the process, where we'd found 5 genes in the pathway. And these genes all encode different types of enzymes, so they're not tandem duplicates of related genes. And each of these enzymes encodes a different step in the pathway. And you can see that the genes are right next to each other. So although this is quite a big stretch, this is 400 kilobases, there are no other predicted genes in between these 5. So it looked as though this was indeed a multi-gene locus, which at the time was a big surprise. We also knew that the fluorescence that we saw in the root tip is actually in the epidermal cell layer. So it's in the outer layer of the root, which is the right place to provide protection against pathogen attack. And when we started to look at the expression of the genes that we characterised using messenger RNA in situ hybridisation, we could see that the genes were expressed in the epidermal cell layer as well. So it looked like the whole pathway is taking place in this cell layer. And here you can see the genes are expressed in the roots, but with little or no expression in other parts of the plant. Many years on, with my friend Bin Han in Shanghai, who does genomics, we sequenced the complete genome of our diploid oat accession, which is actually 4 gigabases. So it's bigger than the human genome. So the oat genome is bigger than the human genome. And we could see, as we expected, we could see similarities with various wheat progenitors, so with other grasses. You can see these blocks of synteny. And here you can see the chromosomes, and our gene for one of the first steps in the pathway is at the end of chromosome 1. And by this point, we'd characterised the entire pathway, which was 12 steps. And this is the pathway from a simple triterpene precursor through to the fluorescent molecule. And we found that there were 12 genes involved, and they're all right next to each other with a tiny little gap here. And so these are the coloured genes. And when you get to either side, the grey genes, those are genes that are not part of the pathway, and they don't have any obvious predicted function in metabolism. And you can see that the genes are expressed in the root tip, as we know, with little or no expression in the other parts of the plant. So this is a weird operon-like biosynthetic gene cluster, and it's located very close to the end of the chromosome, which is here. And very strangely, the genes are in approximately the same order as the biochemical pathway itself, which we could come back to. So this was a big surprise that we'd found the pathway genes to be organised in a cluster. And without going into too much detail, I told you that this molecule is specific to oat. So if we look at the region of chromosome 1 where the cluster is, which is here, and we compare it with alignments to other cereals and grasses, brachypodium, rice, barley, and a triticum genome sequence, again you can see regions of synteny, regions of similarity here, but when we get to the cluster, there's nothing. It's as if it's just inserted into this end of the chromosome. And we also know that if we take the genes within the cluster and we interrogate the genomes of other cereals and grasses to find the closest match, we don't find a really close match. We find some very weak off-target hits, if you like. And you can see that here. So low similarity is in blue. Here we've got, this is again genes along the chromosome. This is the adenosine cluster. And when you go out to the closest matches in these other cereals and grasses, you get very little similarity. But at the flanking regions, there is similarity. And just another way of looking at that is to take the cluster region here, which is in oat. This is just the genes within that cluster, and go out to those weak hits in barley, brachypodium, and rice. And there's no evidence of clustering. So this cluster has come from outer space and inserted into the end of a chromosome of oat, since oats have evolved away from other cereals and grasses. And just before I finish this part, one of the projects I have ongoing is to try to engineer this pathway into wheat to see if we can make wheat disease resistant. So that's a big, ambitious project. And very surprisingly, although the adenosine pathway is specific to oat, and I showed you the genes are expressed in the root tips, if we take the promoters of those genes and hook them up to a reporter gene, in this case Gus, which gives a blue colour, and put those constructs into other plant species, here we've got wheat, but we've also done this with Arabidopsis, with legumes, with other plants, we get the same expression pattern. The promoter behaves itself. And this suggests to us that this oat-specific pathway has plugged into some sort of ancient, highly conserved root developmental process, because the promoters are still reading out, even in disparate species, which actually has practical uses for genetic engineering. So just to come back to the clustering phenomenon, when we started doing this work, it was generally expected, and it was known for some of the very small number of plant natural product pathways that had been characterised at the time, it was expected the genes would be distributed across the genome. But we found this example from oat. There had been one other previous report from maize of a different biosynthetic gene cluster for a different class of plant natural products called hydroxamic acids, which were also involved in defence. But then we and others started to discover more examples of these biosynthetic gene clusters. This is from a review we published in 2014. So these are from Arabidopsis, lotus, tomato, poppy. This is work from Ian Graham's group at York, sorghum, rice, and so on. And just to show you what those structures look like, here's our molecule. This is the maize molecule. And then we found molecules in Arabidopsis that are encoded by biosynthetic gene clusters. And here we've got cyanogenic glycosides in lotus, and alkaloids in poppy, and so on. We then published another review a few years later on, and you won't be able to see this, but my only point is that the list of reported plant biosynthetic gene clusters is continuing to grow, and the chemistry is becoming even more diverse. So this is in 2016. Since then, there have been many more examples reported. So it's not the case that all plant natural product pathways are organised in biosynthetic gene clusters. Some of them clearly aren't, and that includes the carotenoids and the anthocyanins, for example. But carotenoids and anthocyanins are pretty widespread across the plant kingdom. So that's something to think about, and maybe you could talk about this after the lecture, is why should some pathways be organised in biosynthetic gene clusters while others are dispersed throughout the genome? One obvious assumption is that these regions have been introduced by horizontal gene transfer from microbes, but I can reassure you that so far there's no evidence for that for the clusters that have been reported. You might argue that if plants are playing with evolving new pathways for new chemistries, and these chemistries are generally believed to be doing something beneficial to the plant in the environment, in protecting or signalling, if you start to generate pathways that make useful molecules, then maybe there's a benefit to driving the good combinations of those alleles into a biosynthetic gene cluster so that they can be readily co-inherited together. And then another argument, and these are not mutually exclusive, is that if you've got the genes in physical proximity, then you have the opportunity to regulate them not just at the level of transcription factors, but also at the higher level of chromatin modification and nuclear organisation. And that may be important because these specialised metabolites are generally, their biosynthesis is very tightly controlled. They're in specific cell types, or they're induced in response to particular stimuli. And that's likely to be important because it's energy expensive to make the molecules, but also, as I said, they're bioactives, and you really don't want to make them in the wrong cell types at the wrong times. So regardless of the reasons for these clusters, there are practical implications, which is that we can use computational algorithms now to scan through plant genome sequences to look for little barcodes that we think look guilty. They look like they are combinations of genes encoding enzymes with probable functions in specialised metabolism. And we might not know exactly what they do, or we don't for the most part. But we can infer that they may be involved in making chemistry. And so we collaborated with Marnix Medema at the University of Wageningen, who's developed a lot of computational algorithms for mining microbial genomes. One of the algorithms he developed is called anti-smash, and the bacterial and fungal people use that a lot. And it's called anti-smash because it's for finding antibiotic biosynthetic gene clusters. So with Marnix, we developed the first customised version for plants, which he's called Planti-Smash. And this is the output from mining a plant genome. This happens to be oat. Using Planti-Smash, it spits out these predicted clusters with the possible type of compound that they might make. A lot of these are just noise. But this is a broad brushstroke, low-hanging fruit type approach, and the top hit is actually our adenosine gene cluster. So there are quite a few genome sequences out there now, thousands, not the 400,000 for all of the plant species on the planet, but there are large-scale sequencing initiatives ongoing to sequence everything. And there are algorithms like this. So conceptually, there's just going to be a huge ton of data that we need to be able to mine. So then we can put together lists of candidate genes and candidate pathways, but if we can't actually test the function, then it's purely an in silico exercise with a lot of signal-to-noise ratio. So we need to be able to rapidly test what these genes are doing. Like many other groups around the world who work on plant natural products, we do use microbes. We use yeast and E. coli to express some of the genes that we're interested in. But more recently, we've been using a very powerful plant-based system, transient plant expression, which enables us to put the genes that we want to express into agrobacterium. So agrobacterium is the workhorse that will transfer genes that you want to express inside a plant cell, and then the plant does all the work. So what we do is put our genes into expression constructs, put them into agrobacterium, and then infiltrate the agrobacterium cells into the leaves. And the plant that's normally used for this is actually a wild relative of tobacco called Nicotiana benthamiana. And we could discuss later on why this is a very useful host. But if you think of it as just a green canvas that you can paint chemistry on. And the great thing is it's really quick. So within 5 days of infiltrating a leaf, you get your product. Here we've got GFP, and this is actually a fluorescent molecule that we've made. So it's very, very quick. This is at analytical scale. If we want to scale up, we actually have a vacuum infiltration device that the engineering people at John Innes made for us, where we can infiltrate the leaves of 4 to 6 whole plants in one go. So it's still very Heath Robinson, it's hard work, but we can infiltrate hundreds of plants if we want to get a lot of leaf material back. Here you can see the leaves from a single plant that has been infiltrated with agrobacterium containing a GFP construct, and you can see the bright green fluorescence in most of the leaves. And these leaves are not fluorescing because they grew after the infiltration process had been done. So it's very effective. And this is Michael Stevenson, a chemist who until recently was in my lab. He now has his own position at the University of East Anglia, but he, just to show he could do it, infiltrated a few hundred plants to purify a gram of 98% pure triterpene. And this is his proud face, not his happy face. He made a really good video. There's a journal called the Journal of Visualized Experimentation. If you don't know it, it's worth looking it up. It has methods, but it also has videos showing you exactly how to do things. And Michael's starring in one of those. So we've got a deluge of genome sequence information. We have a very rapid way of testing gene function. And we can scale up. So we can scale up to evaluate biological activity, but also to determine the structures of new compounds by NMR, where you need a mig or so to, well, less, but to do structural validation. So coming back to the triterpenoids, the great thing about them is they all come from one linear precursor, 2,3-oxidosqualene. And 2,3-oxidosqualene is widespread. It's in your bodies, it's in plants, it's present across the kingdoms, because it's a precursor for essential sterols. So 2,3-oxidosqualene is used to make the sterols that we have in our bodies, but in plants it's used to make structural sterols such as cycloartenol and steroid hormones. It can also be cyclized to other scaffolds, so it's like a complex origami process. And I'm just showing a few here, but there are over 200 different scaffolds reported so far. And the enzymes that do this are called triterpene synthases. They're also known as oxidosqualene cyclases. So this enzyme family takes one substrate, does an origami thing, and can spit out over 200 different cyclized products. So that in itself generates a lot of diversity. These are steroid or steroid-like molecules. These can then be selectively oxygenated, usually by cytochrome P450 enzymes. And these are the kinds of modifications you can't do with chemistry, or at least for the most part, because you can't selectively modify the different parts of the ring in this way. So this is the power of enzymes. And these oxygenated groups are handles that can then be used to attach other modifications, which may be sugars or acyl groups and so on. So enormous structural diversity from one precursor. The triterpenoids include the steroids, and they are a very rich source of pharmaceutically relevant bioactives. So we then did a proof of concept where we took a triterpene synthase that makes the most common triterpene scaffold, known as beta-amarin. That's the one that Michael made 98% pure, 1 gram of. And then we took 5 cytochrome P450s that had been previously shown to selectively oxygenate the scaffold at different positions. And then we simply co-infiltrated agrobacterium strains containing expression constructs for this and each of these in pairs, and then we factored in combinations of P450s. And just with these 6 starting enzymes, we made over 40 different selectively oxygenated beta-amarin derivatives. We were mixing genes from across the plant kingdom, so some of the molecules we found had previously been reported and others were new to nature. And then James Reed, who did this work during his PhD, purified 9 of these analogues, and in collaboration with our colleague Maria O'Connell at the School of Pharmacy at UEA, he tested the anti-proliferative activity of these molecules against human cell lines and also looked at their the TNF-alpha anti-inflammatory properties. And so this is just a proof of concept to show that we can access the chemistry and we can do that in sufficient quantities to look at structure-activity relationships. We then went back to our pathway from oat, which is reasonably complicated, and we assembled that in Nicotiana, again by transient expression. So this is a pathway normally expressed in the roots of oat, and we're now expressing it in the leaves of a eudicot, Nicotiana, and it works. And here you can see the fluorescence of the leaves because we're making the fluorescent adenosine molecule. So it's a very flexible and powerful approach. So then we got a bit too overconfident and thought, well, we've done adenosine, but this molecule, which is produced by the Chilean tree that I mentioned, also so Chiaja saponaria, the soapbark tree, this grows in the central part of Chile, and this molecule, which is a more complicated triterpenoid, is very important as an immunostimulant. It's extracted from the bark of the trees, so the trees need to be 25 years old. Stripping the bark off is not good for the tree. QS21 is purified from the bark extract, and it's currently worth between $100,000 and $400,000 a gram because it's used in human vaccines for shingles, malaria, COVID, respiratory virus, another respiratory virus, and a whole ton of other vaccines in the pipeline. So there's concern about the stability and the environmental sustainability of supply of this molecule. And so the other point about this is it has a lot of very interesting structural features, different sugars, a very weird C18 acyl chain, which is only present in this molecule out of all the molecules reported from nature so far. And which is important for its immunostimulant activity. So we decided we would try to elucidate the pathway for this molecule. So just to compare again, here's the adenosine with the beta-amarin scaffold, oxygenated groups, 3 sugars here, and the fluorescent N-methylanthrinolate group here. Here's QS21. It's actually got the same core scaffold as beta-amarin, as adenosine, which is beta-amarin. It's got 2 sugar chains in green, and this weird acyl group, and then another sugar. So we started by using publicly available resources. And there's a project, it's quite old now, 2019, it was before then, the 1KP project. I don't know how many of you have heard of the 1KP project? A few. It's one of those really simplistic things. It's a great community effort. It's called 1KP because the ambition was to generate transcriptome sequences for 1,000 plant species. And so it's a fantastic resource. It has its limitations because in some cases it might be a whole seedling that was used, and in other cases it might be the leaves and so on. But it's a very good first port of call if you want to look to see if you can find genes that you're interested in. And there was transcriptome data in the 1KP project for our Chilean tree. And so we'd got pretty good at identifying beta-amarin synthases, which are the genes encoding the enzymes that make this scaffold. Beyond that, we knew that there were oxygenation steps. And we inferred, based on what we've learned from working on other triterpene pathways, we had a rough idea of what types of P450s would do these two steps. And so we managed to predict candidate genes, and we started to piece these together and express them by transient expression using our plant system. So we managed to get to queleic acid. The first three, as I say, were mainly candidate gene-based approaches. This one was more problematic, and we had to do a bit of sifting and removing things that we thought were irrelevant. But eventually we got to queleic acid, which is the oxygenated scaffold. But it's still a long way from queleic acid to this. So we then decided that we needed to get hold of the plant material. So this is James Reed in the lab, and this is the emotional day when the tree arrived. And again, how many of you know about the Nagoya Protocol and the Biodiversity Convention? Not many hands going up there. So this is about responsible access and benefit sharing, responsibly sourcing material. So we didn't go to Chile and rip this sapling out of the forest and bring it back. We did a lot of research to find out where we could source it from, so that we could show the provenance of the tree, we could show where we got it from, and we could show that we hadn't done anything by way of biopiracy, if you like. It turned out that there was a nursery in Cornwall that had been growing these trees for very many years, and they had a paper trail that went back 20 or so years. So we were able to get hold of the tree. This is really, really important. Whatever you work on in the future, whether it's bacteria, plants, animals, you need to understand this because it is a major topic of discussion. So we sequenced the genome of the tree, and this is a much smaller genome than the oat genome, 411 megabases, and we did the karyotyping. So this then gave us, we also did generated transcriptome data for multiple tissues of the tree. So this gave us our own in-house resources, and critically we also had the tree from which we generated all the sequence data, so everything matched up. So then we could start looking for downstream pathway steps. So we already had the 4 that I mentioned, and when we started to look at transcriptome data from different tissues, we found that those 4 were highly expressed in the primordia, so that was useful. And we could look for genes that were co-expressed with these. So we could use statistics to look for genes that were significantly co-expressed with the early pathway steps, because we expected the genes of the pathway to be coordinately regulated. And I've also mentioned to you the idea of biosynthetic gene clusters, and so we could use Plantismash to mine the genome to look for candidate biosynthetic gene clusters. And so we used a combination of approaches to start to look for the next few steps. We knew, based on our knowledge of triterpenes and what had been reported from nature, that the next steps beyond the oxygenated scaffold were likely to be the addition of sugars on the left-hand side of the molecule. And so we were looking for sugar transferases. And there's a particular family of sugar transferases called UD-dependent glycosyl transferases. And we started to prioritize those using our sequence resources. And this was our hit list of top candidates. And then we could do our co-expression with the genes we'd previously characterized and start to piece together the different steps that add the sugars. And so now we've gone from queleic acid to adding on the trisaccharide chain here. And it turns out there are 2 different flavors of trisaccharide chain. So QS21 actually has structural variants. So we got to this point, and I should say that nothing was known about this biosynthetic pathway at all when we started this work. And so at each step we were doing large-scale vacuum infiltration and purification, so we could do NMR to confirm the structure of the products. So these are the genes in our hit list that we identified as being important for this. So again, just to reiterate the speed with which we can go through this process, 5 days from leaf infiltration to working out what we've made, rapid small-scale function analysis, and then we can scale up when we need to. And this was critical for this work. So I had a very talented natural product chemist in the lab, Amma El Demadash, who purified all of the intermediates, and then we started to look at the right-hand side of the molecule. There's a sugar chain here. And he was purifying compounds in milligrams to tens of milligrams amounts and determining the structures empirically de novo, which was a huge piece of work. We got a bit stuck. I've told you about the left-hand side here. We'd already got the steps for this side, but we were worried because the levels were very, very low and we didn't think we'd be able to get through to finding the steps needed for the addition of the acyl chain and the final sugar. And what we found out, and this is an unexpected serendipitous discovery, one of the sort of best scientific discoveries, this sugar here, the first sugar in this sugar chain, is D-fucose. And the only reason I'm saying that is because D-fucose is actually very rare. And it wasn't known where D-fucose came from in plants. The other sugars we knew most of them are available as UDP sugar donors, which can be taken by, so those are activated sugar donors that are present in plant cells that can then be taken by the sugar transferases and used as substrates to add to a scaffold. But nothing was known about where D-fucose came from, and we couldn't find any UDP D-fucose in our plants. And then it turned out that the sugar transferase that added what we thought was D-fucose, which is here in a biosynthetic gene cluster, was right next to a gene for a predicted reductase. And the reductase was tightly co-expressed with the sugar transferase. And it turns out that the sugar that's added here is not D-fucose at all. It's a precursor of D-fucose, which is added to the scaffold and is then reduced by this reductase to give D-fucose. So this, off the back of this, we got a science paper. This is very important, and it's likely to have broader implications for other plant compounds, including drugs and medicines. So if it hadn't have been for this clustering phenomenon, we would never have found this. This meant we could then move forward because we'd boosted the amount of the defucosylated intermediate substantially using this reductase. So then we could go on and look for the further steps. By this point, the people in the lab were mixing a huge number of agro strains, and you just needed one not to grow and you couldn't do the infiltration. So then we decided we needed to start taking the genes that we'd characterized and making multi-gene constructs. And we did this using Golden Gate, which is a modular cloning system. So this meant that we cut down the number of agrobacterium strains we needed to grow to express all of the characterized steps that we had. And then we went after the steps for this unusual acyl chain here and this missing sugar. So we had no idea where the acyl chain came from, but we took a hypothesis-based approach and predicted various classes of enzymes that we thought would be involved. And then we went back to our sequence resources. So this is really interesting. So most diploid plants have around 30,000 genes. Arabidopsis has around 30,000, and so does the soapbark tree, even though it's much bigger than Arabidopsis. And because we'd learned a lot from our computational approaches, we developed a way of ranking candidate genes that was more sophisticated than our earlier approaches. And we used 4 different parameters. I told you that the genes are generally expressed at high levels in the primordia. The genes are generally co-expressed with other previously characterized pathway genes. If the genes are in a biosynthetic gene cluster with a previously characterized pathway gene, that's an extra weighting. And then if they're just in a biosynthetic gene cluster but with no characterized genes, that's another measure. So Charlotte Owen, who's a bioinformatician in the group, devised a way of integrating those 4 readouts. And this is what she came up with. So out of the 30,000 genes in the soapbark genome, this is a hit list of the top 50. The ones in grey are genes that we'd already characterized as part of the earlier part of the pathway. The ones in colours are the genes belonging to, predicted to encode classes of enzyme that we thought may be involved in the addition of the acyl chain. So we had all of the genes necessary to make this advanced precursor, and I won't go into this, but we'd worked out how to make the C9, so the acyl chain is C18 and it comes from 2 acyl units like this. So we had the genes for those. And what we did was we took the genes for both of these components, and then Letitia Martin in the lab said, well, I've got a load of candidate genes here, why don't I just throw them all in together? So I callously laughed at her. So she put over 70 agrobacterium strains into Nicotiana benthamiana, and she got a little peak, and the mass of that was the mass consistent with QS21. So this really is a shotgun approach where how on earth it works, how you get 70 or so agrobacterium strains to coordinately transform and express in this process is quite amazing. So Letitia then went on and deconvoluted the batch of candidates that she had infiltrated to find the 5 missing steps needed for completion of the QS21 pathway. So the soapbark tree makes not only QS21, which is historically called QS21 because it was fraction 21 of a saponin fractionation, it makes over 100 different structurally related molecules. QS21 is currently used in human vaccines, but that's because it's the most abundant immunostimulant saponin in the bark extract, but it also has a degree of toxicity towards humans. There are other molecules such as QS7, which we've also made, which is a simpler version. It's a very effective immunostimulant and it's less toxic. So now we have the genome sequence of the tree, we have the transcriptome resources, and we have a way of making these molecules free from tree. This opens up the way to make both known and new to nature saponins with a view to developing the next generation of vaccine adjuvants. So just a timeline here. The adenosine pathway, which I told you about, took forever. It took from the 1990s to 2020 something and involved a lot of people. The soapbark project, we started in 2017 by getting a small amount of funding from John Innes to do some transcriptome work. Then I got a BBSRC grant, which was only for 2 years, but then Big Pharma got to find out what we were doing and put over £2.5 million into the lab to complete the elucidation of the QS21 pathway. So we went from 2017 to basically completing the science in 2021, so that's faster. And off the back of that, we got another grant, I got a welcome award, and we've spun out a company. But my point, which I'll make in a minute, is that this whole process is getting faster and faster and faster, so the more you learn, the faster you can go. So this slide is out of date, for which I apologise, but the number of plant genome sequences available is now several thousand. But as I said, there are 400,000 species of higher plant on the planet, and in fact new species are being discovered every year. I think you might hear about that tomorrow. And there are these major large-scale sequencing initiatives. There's the Earth Biogenome Project, which is an international project, and within the UK, the Darwin Tree of Life Project. So these are all aiming to sequence just about everything. And as I said, plant genomes contain the instruction manual to make the enormous array of chemistry that is out there in the plant kingdom, most of which we haven't yet discovered. So we work not only on oat and soapbark, we've also worked extensively on wheat, looking at defense-related compounds. We've worked on the Brassicaceae. This is a member of the Caryophyllaceae, so it's related to the pink family. It grows in the UK. It's actually quite invasive. It's called soapwort. So the soapbark tree is taxonomically different. Soapwort, again, the reason why they have soap in their names is because traditionally they've been used as sources of natural soaps. So soapwort used to be grown in the UK by the rivers near the wool mills because extract from soapwort was used as a gentle soap for washing wool with. We've worked on the neem tree and its relatives, the chinaberry tree, because they produce bee-friendly anti-insect compounds. We've worked extensively on Arabidopsis, which produces triterpenes that go out into the soil and shape the composition of the microbial communities around the root. And we've even worked on sea cucumber, which is one of the few animals that actually makes its own defense compounds. And this is a bowl of sea cucumber soup that I ate in Dalian in China, so the compounds didn't protect it. So the other thing I should say actually is, so for soapwort, my student Jenny Jo elucidated an entire pathway which is nearly 20 steps. And very strangely, this molecule actually makes molecules that are very closely related to QS21, which is bizarre because these species are taxonomically disparate. But she elucidated this whole pathway within her PhD, basically within 3 years because she was a rotation student. And it wasn't because she could take the genes from soap bark and look for homologs in soap wort, because they're so taxonomically different, you can't do that. So she had to do the whole thing de novo. And the only slight cheat that she had was that she could use the purified intermediates from soap bark to check whether she was making the same compounds or not. So again, my point is that things are getting faster and faster. And we can move away from thinking about individual pathways to taking a larger scale approach. I've talked about mixing and matching genes from across the plant kingdom to do combinatorial biosynthesis. This is something the microbial world has been doing for a long time. But actually, for microbes, it can be much harder to take a gene from one bacterium and put it into another one. They tend not to work. Whereas because higher plants have a much more recent, narrower evolutionary base, it's a lot more flexible. You can mix and match across the plant kingdom. Where there are multiple genomes available for closely related species, we can start to look at different flavours of things. These are just looking at variants of biosynthetic gene clusters across the Brassicaceae, where we found that there is a palette of, so this is an oxidosqualene cyclase, triterpene synthase family, a subset of P450s, and a certain type of acyl transferase that are making different flavours of acylated triterpenes. And we're also using machine learning-based approaches to try to predict enzyme function from DNA sequence. This is the triterpene synthase family. We're now drawing trees with 13,000 triterpene synthases. So there's a huge amount of metadata out there which is going to grow. Curating it and working with it is challenging. I asked the group to go through the freezers to find out what we had, because we'd purified so many new molecules. These are not molecules you can buy. You know, we can make them, but you can't buy them. And we put together a little library of compounds where we had a reasonable amount that we'd generated over the years from different projects, mainly triterpenes, but not all. And then we put a batch of, I think, 32 of them. We sent them to a company in Germany called OncoLead, who tested them against 90-something different cancer cell lines. And you can just about see the readouts here. And the only point is you see differences. But the thing is, we can access the chemistry, we can start to work with it. And the aim is to try to understand structure-activity relationships. So in the 19th century, advances in chemistry led to the beginnings of the pharmaceutical industry. But now we're in, it's not the beginning of the biorevolution, we're into it, the 21st century, where we have all of this digital input, these DNA sequences, this instruction manual. We have engineering biology, synthetic biology. We can standardize, we can get genes synthesized, you can automate or outsource all of that. We have rapid transient expression. We need to work on expanding our ability to do large-scale analytical chemistry with these types of compounds for which there are no standards in the databases. We can do large-scale expression and purification and NMR. We can build, I mentioned machine learning and AI, and of course that covers a huge area. We can use machine learning-based tools to try to predict enzyme function from DNA sequence. But one of the other things we're doing is training models to learn the structural features of molecules that are associated with particular types of bioactivity. So there are big publicly available databases out there like PubChem, where you can look at data for huge numbers of molecules against a particular bioassay, and then you can train a machine learning model to tell you what the desirable structural features of those molecules are and the undesirable ones, and use that to design new molecules. And so our ambition, which is ridiculously big, is to go round and round this loop, learning more and more about how to make molecules and how to refine their structures so that we can ultimately make designer bioactives for particular applications. So I'd like to finish there just by drawing your attention to the many people who've contributed to this work, both in my lab and our collaborators, and also our funding bodies. And I should just point out that we have a spin-out company which celebrated its first birthday yesterday, called hothouse therapeutics, which is currently in my lab, but will move out at some point. Thank you.